I just have one of these golf pencils.
The sad thing is, we have all of these. I need to go next to the other guy.
All right, guys, so thank you so much for coming to the workshop. The hope is that we'll start doing this a little more often, so definitely look forward to future workshops as well. Like all of these workshops, though, the goal is always that you'll be able to pass the content assessment. In order for you to do that, it's really important that we are participating 100%, which means you're taking notes, answering questions. Also, if you have questions, please make sure you ask those questions, and I will do my best to answer them as well. But this should last about 35, 40 minutes, and it'll be pretty interactive as well. All right, so the thing I always like to do when I go to a playlist is start with the objectives. And so I just copy and pasted the list of the objectives for each of the playlists. And so kind of think about these objectives as we go through this workshop. And so the very first objective for DNA to protein is to compare the structures of DNA and RNA in order to identify similarities and differences. Okay. And so after we finish this first section, you're going to just basically record the answer to this objective or describe the differences between DNA and RNA. And so kind of just keep that in mind as we do this. All right,
you better catch it.
I really like this image a lot because this image really encompasses everything that you need to know on the DNA to protein playlist. We start up here with the DNA. We go through the process of transcription to make RNA, and then we go through the process of translation to turn that RNA into proteins. And this image will make a lot more sense to you by the end of the workshop, but also kind of keep this image in your mind as you go through this content. Okay. So let's start out with DNA and RNA and kind of compare them side by side. So here we have DNA, here we have RNA. Look at the pictures right now and try to identify some similarities and differences between DNA and RNA. What are some things that stand out to you guys? Maybe just take a couple seconds to think it over. What are some differences you notice between the picture of the DNA and the RNA? Here you go, Jackie.
Can you finish this?
Yeah, that's one of the really important differences. Notice how DNA has 2 strands, RNA only has 1. So we say DNA is double-stranded, RNA is single-stranded. Okay? What other differences do you notice? Really pay attention to the details.
Well, I mean, it's true because I mean that it can be negative too.
If you think that's true, then
It's pretty easy.
Anything you guys notice that's different about the letters in the DNA versus the RNA?
Or like, can you open my two as soon as I request it? Which is like
Yes, did everyone hear what Alondra said? DNA has the D, RNA has the R. And this is the type of sugar that's in the DNA and the type of sugar that's in the RNA. DNA has a deoxyribose, that's the D in DNA. The RNA has ribose, which is the R in RNA. And so that's the other big difference between DNA and RNA. And there's one more difference when you look at the bases. You notice anything different about the bases in RNA versus the bases in DNA? Yeah, so instead of a T in DNA, which is thymine, we have a U, which is uracil. Okay? So these are the 3 big differences between DNA and RNA. And so let's take some notes about that right now. So the first big difference that we talked about, DNA is double-stranded. There's 2 parts to it, 2 sides to it. RNA is single-stranded. There's only 1 side to it. So make sure you're writing that into your notes. So make sure you write that into your notes right here. All right. Do you need a pen or pencil? All right, great. The second big difference, like Jackie said, DNA uses thymine or T, so you'll see some T bases inside the DNA. There's no T's in RNA. RNA actually uses something called U or uracil, and so you'll see there's some U's in this RNA. That's the second big difference. Record that in your notes as well. And the last big difference, like Alondra pointed out, the sugar in DNA is deoxyribose. That's why there's D's on these sugars. And the sugar in RNA is ribose. That's why these are R's. So these are the 3 big differences between DNA and RNA. So definitely know these like the back of your hand. Know the key differences because there'll be several questions on the content assessment asking you about these differences. You guys all get that done? All right. This is just some extra information, but this is a kind of a chemical formula of the deoxyribose sugar, the ribose sugar. Notice that the only difference between the sugars is the addition of this little O here in the ribose. That O, or the oxygen, is missing in the deoxyribose. It's not something you need to know, just that's how these sugars are. All right. So what I want you guys now to do is we have some DNA right here. This is a practice problem. I want you to draw what the RNA would look like in your notes. And so draw the structure of the RNA and just make sure you label the different parts. And then put how it would be different. So in RNA, we know that there's an A base. But like for example, we know there's not T bases, so draw what that T base would be. All right. Also keep in mind that the sugar in DNA is a deoxyribose, so draw what the correct sugar would be for the sugar in RNA. So finish drawing it, but make it for the RNA.
Not right now. We'll play this stuff.
Okay.
DNA protein. Can you pause it?
This is one mistake on my part, but you actually want to get the base that matches the A. So this A actually matches with a U in RNA. So make sure you correctly label that. So A's go with U's, G's go with C's, T's go with A's, C's go with G's. So make sure you put the correct base that matches it, sorry. So it's not an exact copy. You almost want to get the one that matches that one. If that makes sense.
No, it wasn't for
So you should be adding the extra bases in. We know that A matches with a T, a U. We know that G matches with a C base or cytosine. We know that T matches with an A base. And so on. So once again, the key thing is just illustrating the differences. We have uracils instead of thymines, and we have deoxyribose instead of ribose. And the last thing I want you guys to know about DNA and RNA, this is more a review of something from the DNA structure of DNA codes, is that just remember this vocabulary word, nucleotides. Because both DNA and RNA are made up of nucleotides. A nucleotide is just a vocabulary word that refers to all three parts of the DNA, the phosphates, the sugars, and the bases. And so this is a nucleotide right here because it has a phosphate, sugar, and a base. And so we have DNA and RNA are made up of nucleotides. So just know that vocabulary word. That refers to the combination of a phosphate, sugar, and a base together. All right, moving on. We're going to do a multiple choice practice problem because I think it's always a good idea to practice multiple choice questions. So the first one, the question is asking, the molecules that contain genetic information of living organisms are DNA and RNA. So these are the keywords, DNA and RNA. Which of the following is a major difference? This is another key word in the question. We want to talk about what's different, not what's similar, what's different. All right, so read these different answer choices and try to find the right answer for that question. So let's go through these answers. It says DNA is a nucleic acid, RNA is a protein. We haven't really talked about nucleic acids or proteins at all, so that's probably not the right answer. So we can probably eliminate that choice. Nucleic acid is also just another word for nucleotide. But we also know that RNA is also a nucleotide, it's not a protein. Okay, so that's definitely the wrong answer. DNA creates proteins, RNA creates enzymes. Okay, so once again, we haven't really talked that much about enzymes, so I think this must not be the right answer choice. We can probably eliminate that. DNA contains uracil, RNA contains thymine. This seems like it might be familiar, but I think it's actually the other way around. I think DNA contains thymine and RNA contains uracil. So this is like a trick answer. So make sure you eliminate this choice because that one's not the right answer. And the last answer is D, which is our correct answer. DNA is double, a double helix or double stranded. RNA is single helix or single stranded. We know this is one of the key differences, so the answer choice is D. So next question, number 4. What is the strand of RNA that is transcribed from DNA with the sequence AGA, TGC, ATC? And the key word for this one is transcribed. And you'll know what this word means more in a little bit. But what that really means is that you're not going to just match it directly. What you want to do is you want to get the bases that pair up with each of these. So think about, you know that A matches up with U, G matches up with C, A matches up with U. And so put in the bases that actually pair up with those bases, so that way you get the right answer. But spend some time filling in the bases that pair up with those to figure out what the right answer is going to be. All right, it looks like most of you guys have this as well. So you want to match it up. Because it says transcribed, you want to match them up. UACG. If we already have enough information, UCU. Okay, the only one that says starts with UCU is A. So A is our correct answer. So let's go ahead and move on. So we have now finished the first objective. We're going to move on to the second objective. Before we do that, though, I actually want you to take some time and write in your notes the differences and similarities between DNA and RNA. So use that objective. Kind of treat the objective as like a question. Like turn this into a question. So like if I was to turn this objective into what are the similarities and differences between DNA and RNA, I want you to write those down in your margin. So in your notes, write down what are the differences between DNA and RNA. So what are the similarities and differences between DNA and RNA? And just write that in your margin. So that way you can kind of use this objective like a quiz question. So go back through your notes if you need to, but you want to write in what are the differences between DNA and RNA, and use this objective like a quiz question. Gosh, up there. Okay. Cool. Nice, like what you guys have so far. You need to go back and check those notes. That's the key one, though, as far as the differences between DNA and RNA. Looks good, guys. Really like what I see being written down. the three big differences are thymine. Let's talk about DNA versus RNA. We're talking about thymine or T. RNA is uracil or U. DNA is double-stranded. RNA is single-stranded. And then the last one is the sugar. So DNA has deoxyribose, which is the D in DNA. RNA has ribose, which is the R in RNA. Okay. Lots of similarities as well. They both have adenine, they both have guanine, they both have cytosine, but these are the three really important differences. All right, so let's go ahead and move on to the second objective, which is describe what happens and the location of each step of transcription. All right, so now that we're moving on, it's important to ask the question, all right, so we have DNA. We know that DNA is like the instructions for everything in our body. Why do we even need RNA? Like, why not just use the DNA to make our proteins and make stuff in our body? And here's why. We know that DNA contains the instructions for protein, and that proteins are made by ribosomes. So we know the ribosomes are out here. In the cell, that's where the proteins are made. The DNA is inside the nucleus. It's actually really risky or dangerous for the DNA to leave the nucleus. We gotta think about the DNA is precious. It's important that we keep it safe. If it gets damaged, then our body can no longer make proteins. So we want to make sure that the DNA is safe inside the nucleus. But the ribosomes, they're really stuck inside the cytoplasm. It's not like the ribosomes can move to the DNA, where the DNA is. They're kind of stuck out here inside the cytoplasm. And so this is why we need RNA. RNA is the messenger. So think of like those owls in Harry Potter. They send messages back and forth. The owl, the RNA, the messenger RNA or mRNA, is a message that can be sent from the nucleus to the ribosomes. And this, so write down this information down here in your notes, that messenger RNA or mRNA. And once again, think of this image of the owls in Harry Potter. It's a message being sent from the DNA, from the nucleus, to the ribosomes out in the cytoplasm. Okay. So there's another vocab word that you want to know. This is the vocab word of transcription. And transcription is basically the process in which nucleotides of DNA are copied into mRNA. And so here we have the DNA. We're using this DNA to make that complementary mRNA or messenger RNA. Make sure you're following along in the notes. So move on to the next page. Still on the right words for that. All right, great. So let's talk about how this process of transcription, of turning our DNA into RNA, actually happens. Okay? So the very first step is that we copy the DNA info onto the mRNA. Okay? And so what we're going to do is we're actually going to write in the corresponding RNA strand. So if this is our DNA, we know that T matches up with A matches up with U, A matches up with U. And we're going to end up with the RNA that complements the DNA. And so notice AT, AU, AU, GC, CG, TA. Very similar to matching up DNA when we do DNA replication. The big difference, remember though, is that there's those U's in the RNA. This is our RNA right here. This is our DNA right here. So very similar, but RNA has the U's. One question I get a lot from students is like, oh, but if there's a T in RNA, what does that match it up? A T still matches up with an A in mRNA. It's just the A matches up with a U in mRNA. So just know that difference when you're doing this as well. All right, you don't have to copy this all down. It's more just to get the general idea of which ones match up. The next step is now that we've made our messenger RNA, our mRNA, that little owl or that messenger is ready to leave, and so we have the DNA inside the nucleus. That messenger is now going to come out and go to the ribosome. So it's leaving the nucleus. I have this great animation I'll show you guys in a little bit that actually shows this whole process in action. But the mRNA is now going to leave the nucleus. Okay. Here's that animation I was telling you about. So here we have an enzyme. We're going to make the mRNA that matches it up. So notice how the G's and C's are matching up, the T's and A's, the U's and A's are matching up. And so now we have our mRNA that's ready to be sent out. Okay? So watch this animation. This is actually how fast it happens. So I'm going to start this actually probably at about 46 seconds.
Yeah.
But this is pretty incredible. This is actually the mRNA being made. And so you can see this red part is the DNA. This is the enzyme making the mRNA. And this big yellow strand, this is the mRNA being made. And it happens super fast. Like, you can see how quickly it's happening.
It's in white.
And so this big long strip of mRNA is being made. And you can notice that really notice that it's single-stranded here versus this dRNA is a lot bigger. It's that double strand. All right. So let's do a practice problem now. Which of the following statements is correct? So this is a key vocabulary word. We're talking about correct, not incorrect, correct. Concerning transcription. So that's another key vocabulary word in this question.
Where is it?
So if you need to, go back and check your notes. But read over which of these 4 statements is true or correct about transcription.
I think it's because we have different classes. We found that one's more advanced for me. They should be the same. Should be the same. They should be the same. But people like the same procedure.
All right. So during the transcription, the adenine of DNA joins with the thymine of the RNA. We know this is not right because we know that actually if we're talking about uracil in RNA, there's no thymine or T in RNA, so we can eliminate that answer choice. During transcription, DNA is used to form a strand of mRNA. Okay, that seems like a possibility. Let's leave that as a possible answer choice. During transcription, both strands of DNA are transcribed at the same time. But that video we just saw, was it both strands being transcribed, or was it just one of them making that single strand? So we know that actually it's not both, it's just one strand. So this is not the answer choice. During transcription, one strand is the leading strand, one strand is the lagging strand. This is a what? I've never heard of this before. That's a clue. If you're getting vocabulary words that you've never heard of before, and you've really studied the playlist, that's actually a sign that that is not an answer choice, because if it was an important vocabulary word, we would actually give it to you. So don't fall into the trap of saying, oh my gosh, this is a vocabulary word I don't know. That must be the answer. This is actually not the answer. This one that we had before, B, is the best answer choice. And so be careful of falling into that trap of finding a vocabulary word that we've never seen before. That may be not be the right answer. All right, so we're done with the second objective. We're about to move on to the third objective. Just like last time, though, I want to turn this objective into a question that you answer. So it says, describe what happens in the location of each step of transcription. So let's make this into a question. What happens? Okay. And we want to know the location and in each step of transcription. Okay? So write down the location of each step of transcription and what's happening. If you need to, go back and check those notes. But I want you to turn this objective into a question and make sure you can answer this question.
Step 7. Biology. Step 7 of biology.
Yes. That's the answer. Okay. So once again, we're turning this into a question. What happens and what's the location of each step of transcription? The location, if you remember, the whole reason why we need mRNA is that the location is the nucleus. This is the part of the cell where this is happening, and eventually that mRNA is going to leave the nucleus. But this is why we need mRNA in the first place, is that DNA can't leave the nucleus, so it has to happen in the nucleus. And during each step of transcription, the big thing that we're doing is we're making mRNA, and then the next step is that mRNA leaves the nucleus. Those are the two big steps of transcription. We make the mRNA, we make that yellow snaky mRNA, and then that mRNA will then leave the nucleus. Okay. Let's go ahead and move on to the third objective. Describe what happens and the location of each step of translation. So it sounds similar than transcription, but there's a big difference between translation and transcription. Let's talk about what that is right now. Okay. Translation is basically the last step of making DNA into a protein. We've made our mRNA. Now we're going to use the process of translation to turn that mRNA into a protein. Okay. So translation is the process in which mRNA is decoded. We'll talk about what decoded means, but this is basically like figuring out the code. Figure out the code in mRNA. Okay? So we'll talk about how it's kind of like a secret or hidden code in the mRNA, and we'll talk about how we figure that code out. And then by tRNA. So tRNA is actually a different type of RNA. So notice mRNA is messenger RNA, tRNA is called transfer RNA, and we'll talk about that later. And we're making a chain of amino acids or a protein. Okay? Okay. So let's talk about how this process actually happens. So step 3. Our mRNA has now left the nucleus, and now this mRNA, right here, this strip of mRNA, is going to attach to a ribosome. So these two circles right here, this is our ribosome, we have our mRNA, and they always attach at a very specific point. They attach at the place where it says AUG, which is called the start codon.
So, you're looking at the rest of
And you might be wondering what this word codon means.
So here you're just answering the same question.
Codon is actually just a sequence of 3 bases. So anytime you see the word codon, just think of 3 bases. Because when we read this code that's in mRNA, it's in groups of 3. So here we have a GCA codon. Before we were talking about an AUG codon. And the reason why we talk about groups of 3 is 3 bases equals 1 amino acid, which equals 1 codon. So add this part to your notes, see if it's not in your notes. 3 bases equals 1 amino acid, which equals 1 codon. I apologize. There's a lot of vocabulary to wrap your head around. That's why you have these notes. I would encourage you to go back and study these notes. But I do think we're also going to post this video online, so you should be able to watch it again if you want to hear this information for a second time. All right. So moving on. This is the chart for deciphering the codons into amino acids. You by no means need to memorize this chart. You do need to know how to use this chart. So let me show you how to use this chart. So we talked about having the AUG codon. This is how we use that chart to figure out what the AUG codon is. So we start with A right here in red. We find U, bring it down here. Inside AUG is the methionine amino acid. And so this is AUG, the code for AUG is methionine. All right, what is UUU? So I start here, U, phenylalanine. Try to find CCC in this chart. CCC. So start with red, move down blue. CCC is proline. What is UAU? UAU, tyrosine. Okay? Once again, you don't need to memorize this chart. You will always get this chart on a quiz when you need it. You just need to know how to use the chart. Okay? So make sure you know how to use it. All right. So this is the code that's inside the DNA, inside the mRNA. All right. So the next step in this process is that our mRNA is attached to the ribosomes. Now we have these things called tRNAs, which are actually the ones that bring in the correct amino acid. They read that code and they say, okay, I have an AUG, I'm going to bring in a methionine amino acid. Oh, I have a UAU, I'm going to bring in a tyrosine amino acid. So it reads that code and figures out what amino acid it needs to bring. Okay? And we've talked about tRNA already a little bit, but the T in tRNA is transfer RNA. And what it just means is it transfers or brings the amino acids to the ribosome. So we have mRNA, which is messenger RNA. That brings the message from the DNA in the nucleus to the ribosome. The tRNA is what reads that message and brings in the correct amino acid to the ribosome. So notice there's differences there. Those are important. All right. The next step, step 5, is now we have brought in amino acids. We're going to kind of connect them together to form a protein. Okay? And so it's kind of hard to see right here in this picture, but essentially individual tRNAs are